talk digestion. So we're going to ignore the mechanical part of digestion. We're strictly going to look at the chemical part of digestion. We break up the chemical part of digestion based off of which macromolecule we're actually talking about: carbohydrates, lipids, or proteins. Chemical digestion of carbohydrates. Proteins are going to only start chemically digesting upon exposure to acidic conditions within the gastric juices of the stomach, and then by enzymes entering the duodenum and through the small intestine. The principal enzymes here: pepsin, trypsin, chymotrypsin. The difference between trypsin and chymotrypsin is that chymotrypsin acts on the acidic part, whereas trypsin doesn't like the acidic part. Carboxypeptidase works on the carboxyl end of the peptide bond, whereas aminopeptidase is going to act on the amine end of the peptide bond. So a peptide bond is the amino acid with the NH2 pointed towards the carboxyl end, COOH, and then they bond right there. So what ends up happening is that carboxypeptidase is going to split on the carboxyl end, whereas the aminopeptidase is going to split on the amine end of that peptide chain. Chymotrypsin elastase is an important enzyme here. It digests elastin. And so what ends up happening is that if we don't digest elastin is going to continue to unwind and unwind and unwind and unwind and unwind. It becomes this infinitely long protein chain that we can't get in and digest all the other proteins that are tied onto the elastin. And this elastin is one of the principal connective tissues in animal proteins, in meats. Elastin and collagen are the two principal connective tissues of skeletal muscle. And so if we don't have an elastinase, something that's going to digest elastin, we're not going to be able to break apart all of the animal stuff we eat. This enzyme here is tied with an evolutionary advantage mutation in one of the lipopolyproteases, the APO gene in particular. Problem is that a mutation in the APO also causes malformation of different types of secreted proteins, in particular tau proteins and amyloid proteins around neurons, and is associated with neurodegenerative diseases. The dipeptidase is going to break up dipeptide bonds. The digestion doesn't take a lot of water to it. However, the absorption takes a lot of water. So people who have high protein diets tend to run the relative risk of increased dehydration, particularly within the alimentary canal, which is why people who have large amounts of animal in their diet or large amounts of protein in their diet need to also consume large amounts of fluid when they eat, because otherwise they run the risk of becoming what? Starts with a C. Constipated. Same thing happens with people who have large amounts of meat and large amounts of cheese but don't have a lot of fiber. So if we look at it in terms of the enzyme breakdown, protein comes in, interacts with the gastric juices, hydrochloric acid, pepsinogen, converting to pepsin. We break down to the polypeptide and the oligopeptides. These polypeptides and oligopeptides are going to interact with pepsin, trypsin, and chymotrypsin to form dipeptides and shorter oligopeptides. These are going to interact with the dipeptidases and the individual amino acidases to break apart into individual components, either into an individual amino acid component or into an individual amino acid itself. Those amino acids will then get absorbed. This happens to every protein and every large polypeptide chain that you ingest and every single amino acid that you ingest, which is why when we talked about things like creatine supplementation, creatine is nothing more than amino acids linked together. If I ingest amino acids, what's the likelihood of me actually ingesting and absorbing that one amino acid as that amino acid? It's very low. The last one we have to look at